All right, welcome to Rad Onc Smart Review, the RadBio edition.
Great to be here.
Today we're tackling a really fundamental topic, absolutely key for understanding radiation biology and honestly for nailing those board exams.
Definitely.
We're talking about how cells handle the most dangerous kind of DNA damage, the double-strand break, where the DNA molecule is completely severed.
Yeah, think of it as snapping the ladder clean in two. And these breaks, they're a huge challenge for the cell.
Yeah.
If they aren't fixed or if they're fixed incorrectly, the consequences are, well, pretty dire. You get mutations, chromosomes getting rearranged, general genomic instability.
Which could lead straight to cell death or worse, cancer.
Exactly. So the cell absolutely has to have good, efficient, and ideally accurate ways to mend these breaks. It's critical for survival.
Okay, so that's the setup. Thankfully, cells aren't defenseless.
Mm-hmm.
They've got two main repair pathways for these double-strand breaks, or DSBs.
That's right.
And for this session, we're going to really dive into the details, the mechanics, the key protein players, and the clinical relevance of these two pathways. Non-homologous end joining NHEJ and homologous recombination repair, or HRR.
We want to pull out all the essential facts, the concepts you really need to have down cold.
Absolutely. So first things first, just really appreciating the scale of this DSB challenge. It's like the most toxic thing radiation does to DNA, right?
It absolutely is the most cytotoxic lesion. So getting that repair right, it's non-negotiable for the cell to survive and keep its genome stable.
And for bird prep, you have to remember that when these pathways fail or make mistakes, that's a major driver for things like radioresistance in tumors or even how cancer starts in the first place.
Precisely. NHEJ and HRR, they're the cell's primary defense team against these breaks.
Okay, so let's start comparing them directly. What are the big differences you need to know? Let's start with fidelity. How accurate are they?
Okay, so NHEJ, it's generally known as the error-prone pathway. Think of it like a quick patch job, maybe a bit messy.
Like cellular duct tape.
Kind of. Its main goal is just speed, get those broken ends stuck back together fast. But that speed comes at a price. You often lose a few bases or maybe gain a few right at the break site.
So you get these little insertions or deletions, indels?
Exactly, small indels. Or sometimes it might even accidentally join two ends that weren't originally together. So accuracy isn't its top priority.
Got it. Quick and dirty, but gets the break sealed. What about HRR?
HRR is the opposite end of the spectrum. It's the high-fidelity pathway. Think like precision engineering. It's designed to be accurate, essentially error-free.
And why is it so accurate? What's the trick?
The trick is it uses a template, an undamaged, identical piece of DNA to guide the repair perfectly.
And in our cells, that template is almost always
the sister chromatid, the identical copy of the chromosome that's made during DNA replication. It uses that sister chromatid as a perfect blueprint to restore the original sequence.
Okay, so that's difference number 2 right there, the template requirement.
Yeah.
NHEJ doesn't need one.
Correct. No template needed for NHEJ. It just grabs the ends it finds. That makes it versatile, available anytime, but it's also why it's less accurate.
Whereas HRR absolutely needs that template.
Absolutely requires it. Primarily the sister chromatid in a million cells. Without that template nearby, HRR just, it can't happen.
And this template thing directly leads to the next key difference when each pathway is active during the cell cycle. This is super high yield for boards.
Oh, totally critical. Because HRR needs that sister chromatid, which only exists after DNA replication.
Right.
It's mainly active only in the late S phase and the G2 phase of the cell cycle.
So after the DNA has been copied, but before the cell divides.
Exactly. In G1, before replication, or in G0, when cells aren't dividing, there's no sister chromatid hanging around. So HRR is pretty much switched off then.
Which leaves NHEJ as the main player in G1 and G0.
Precisely. NHEJ is active throughout the whole cell cycle, G1S and G2. But it's the dominant pathway when that sister template isn't there, so mostly G1 and GO. And like we said, it's generally a much faster process than HRR.
Okay, quick summary before we get into the protein names. NHEJ, error-prone, no template, active all the time, but dominant in G1, G0, and faster.
Yep.
HRR, high fidelity, needs the sister chromatid template, mostly active late SG2, and slower.
You got it. Nailing that cell cycle distinction is crucial.
Okay, let's get specific now. Proteins. Who are the members of the quick fix crew, NHEJ? What happens right when that break occurs? Who shows up first?
So the very first responders on the scene are Q70 and Q80. They bind together, form a heterodimer, like a little ring or clamp.
Q, 70, and 80.
Right. Q7080, think of them as, molecular clamps. They recognize the broken DNA ends almost instantly and bind right onto them.
So they're the sensors. They find the break and grab on.
Exactly. They bind the ends, whether they're blunt or have little overhangs. And this is really important because, one, they protect those fragile ends from getting chewed up by other enzymes.
Okay.
And two, they act like a landing pad, recruiting the next set of proteins to the site.
All right, so Q7080 clamps on who they call in.
They recruit a really big kinase called DNA PKCs. That's DNA-dependent protein kinase catalytic subunit.
DNA PKCs. Okay, that's a mouthful.
Yep.
So Q brings this kinase to the party.
Yep. And binding to the Q-DNA complex actually activates the kinase activity of DNA PKCs.
What does it do once it's active?
Well, the whole Q-DNA PKCs complex helps sort of bridge the two broken ends, holding them close together, synapsing them. But maybe its most critical job is dealing with messy ends.
Right, because breaks from radiation aren't always nice, clean cuts, are they?
Almost never. They're often dirty ends, maybe with damaged bases or weird chemical groups attached or incompatible overhangs. They need processing before they can be stuck back together.
So how does NHEJ handle that? This seems like where errors could creep in.
It is. This is a key step. DNA PKCs phosphorylates and activates another protein called Artemis.
Artemis, okay. What's its job?
Artemis is a nuclease. It cuts DNA. It has both endonuclease activity cutting within a strand and exonuclease activity chewing back from an end. It's the main tool used to trim off those damaged bits or incompatible overhangs, basically making the ends ligatable, ready to be joined.
So it cleans up the ends, but without a template to guide it.
Exactly. That's where you can lose information. There are other helpers, too, like DNA polymerases, MUV, and lambda that can fill in small gaps, and an enzyme called PNKP that cleans up certain chemical end groups. But Artemis is the star player in end processing, regulated by DNA PKCs.
Okay, Q binds, recruits DNA PKCs. DNA PKCs bridges the ends and activates Artemis. Artemis processes the ends. What's the final step? Sticking them together.
That's ligation, sealing the deal. This is done by another protein complex.
More proteins.
Yeah, a core complex of 3. XRCC4, DNA ligase 4, and XLF. XLF is also sometimes called cernunose.
XRCC4, ligase 4, XLF cernunose. Okay, how do they work together?
XRCC4 acts like a scaffold. It holds DNA ligase 4 in the right place and stabilizes it at the break. DNA ligase 4 is the actual ligate. It's the enzyme that reforms the phosphodiester bond, reconnecting the DNA backbone.
And XLF.
XLF sort of stimulates the whole process, helps make the ligation more efficient, especially if the ends are a bit tricky.
Okay, let's recap the NHEJ flow. Q finds and binds ends hens, recruits DNA PKCs. DNA PKCs helps bridge ends and activates Artemis and others. Process the ends. The XRCC4 ligase 4 XLF complex comes in and ligates.
That's the core pathway. And remember that error-prone label, it really comes from that Artemis processing step. Trimming ends without a blueprint inevitably leads to small deletions or maybe small insertions if those polymerases fill things in a bit creatively.
But it's fast and works anytime, especially G1, so it's essential.
Absolutely essential.
Okay, remembering these names for exams. Q, DNA PKCs, Artemis ligase 4. Any tricks? Maybe a mnemonic? How about kids play around loudly?
Hey, I like that. K for Q, P for DNA PKCs. We can say the play around reminds us of PKCs. A for Artemis, and L for ligase 4. Kids play around loudly. Yeah, that could definitely help lock those in.
Great, so that's the quick fix crew. Now let's switch over to the precision engineering team, HRR. We know it's more complex, slower, but accurate, and it needs that sister chromatid, so it's mainly late S and G2.
Right. And the first step, again, after the initial sensing, is quite different. It involves preparing those DNA ends specifically for using a template. A key early sensor and processing complex here is the MRN complex.
MR11, RAD50, and MBS1. Right?
Correct. MR11, RAD50, MBS1. And they team up with another protein called CTT.
Okay, MRN and CTP, what do?
They initiate a really crucial step called resection, specifically 5' to 3' resection. MR11 itself has nucleus activity, and together with CTIP, they start chewing back one of the DNA strands at the break.
Chewing back the 5' end?
Exactly. They chew back the 5' ending strands on both sides of the break. This creates long, single-stranded DNA tails that have a 3' end. These are called 3' overhangs.
And this resection, is this like a point of no return for HRR?
It's a major commitment step. Once you get significant resection, creating these long 3' ssDNA tails, the cell is basically committed to trying HRR. If resection is blocked, the balance tips back towards NHEJ.
Okay, so now we have these long, single-stranded 3' tails. They sound kind of vulnerable. How does the cell handle them?
Good point. They are vulnerable. That's where RPA comes in. Replication protein A.
RPA.
RPA is a 3-subunit protein complex, and it very quickly coats these newly generated single-stranded DNA tails.
Coats them. Why?
For protection, mainly. Protects them from being attacked by nucleuses, smooths out any weird secondary structures they might form, and importantly, keeps them accessible for the next stage of the process.
And this next stage involves some pretty famous proteins, especially in cancer biology.
Absolutely. Now we bring in the heavy hitters, BRCA1, PLB2, and BRCA2. And another protein called Rad52 is also important here.
BRCA1 and BRCA2, the breast cancer genes. And PLB2 links them.
That's right. BRCA1 often gets recruited early. It helps promote that resection step we talked about and acts sort of like a signaling hub. PLB2 acts as a bridge, physically linking BRCA1 to BRCA2, and BRCA2 has a super critical job.
Which is?
Its main role here is to kick RPA off the single-stranded DNA.
Oh, okay. Displace it.
Exactly. Displace RPA and then load the key enzyme for the next step onto that ssDNA, and that enzyme is Rad51. Rad52 also helps with this Rad51 loading process.
Rad51, that sounds important. What does Rad51 do?
Rad51 is the star of the show for HRR. It's the recombinase. It gets loaded onto that 3' ssDNA tail, forming a long filament, a Rad51 ssDNA nuclear protein filament.
And this filament is what actually finds the template.
Precisely. This Rad51 filament has the amazing ability to search through the genome, find the homologous sequence on the sister chromatid, and then invade the double helix of that sister chromatid.
Invade it. How does that work?
It basically forces its way in. The single-stranded tail from the broken DNA pairs up with its complementary sequence on one strand of the sister chromatid, displacing the other strand of the sister chromatid locally. This creates a structure called a D-loop.
A D-loop. Displacement loop. Okay. So now the broken strand is paired up with the template. What happens next?
Now you have a 3' end from the invading strand sitting right next to its template on the sister chromatid. This is a perfect setup for DNA synthesis.
So DNA polymerase comes in.
Exactly. DNA polymerases are recruited. They use that invading 3' end as a primer and the intact sister chromatid strand as the template. They then synthesize new DNA, accurately copying the sequence from the template and effectively filling in the information that was lost at the original break site.
So it's using the blueprint perfectly.
Perfectly. And then there's a whole series of steps involving unwinding enzymes called helicases, and enzymes that resolve the intertwined structures called holiday junctions often form.
Right, resolving those junctions.
Yes. Resolving those junctions, often involving specialized nucleuses. And then finally, any remaining nicks in the DNA backbone are sealed by DNA ligases. The end result is 2 intact, identical DNA molecules with the original sequence perfectly restored at the break site.
Wow. Okay, so let's trace that HRR pathway again. mRNC type E start resection, creating 3-foot tails. RPA coats the ssDNA, BRCA1, polysoll BRCA2, and Rad52, displace RPA, and load Rad51. Rad51 forms a filament, finds the sister chromatid, and invades, forming a D-loop. DNA polymerases synthesize new DNA using the sister chromatid as a template. Complex resolution steps, holiday junctions, etc., and final ligation.
That's the essence of it. Much more complex than NHEJ, definitely slower, but that complexity buys you the accuracy. It's absolutely vital for maintaining the genome, especially when the cell is getting ready to divide.
Okay, and this is where it gets incredibly relevant for clinical practice and definitely for your exams. Mutations in BRCA1 and BRCA2.
Absolutely critical. You have to know this. Mutations in BRCA1 and BRCA2 dramatically increase the risk of developing certain cancers, breast, ovarian, prostate, pancreatic, the list goes on.
And that's directly because of their role in HRR.
Directly, they are quintessential tumor suppressor genes. If a cell has a faulty BRCA1 or BRCA2 gene, its ability to perform high-fidelity HRR is severely compromised. It loses that precision repair mechanism.
So what happens then? How does that lead to cancer?
Well, the cell still gets DNA damage, including DSBs. Without efficient HRR, it's forced to rely much more heavily on the other pathways, particularly the error-prone NHEJ pathway we just discussed.
So it's making more mistakes when fixing breaks.
Exactly. More mistakes, more mutations, more genomic instability, and that accumulation of genetic errors is what drives cancer development.
And this defect, this HRR deficiency caused by BRCA mutations, is precisely what makes those cancer cells vulnerable to a specific class of drugs, PRP inhibitors.
Yes. This is the classic example, the poster child, for a concept called synthetic lethality. You absolutely need to understand this for boards and for practice.
Okay, break down synthetic lethality for us in this context. PRP inhibitors. They target PRP1, right, which is involved in repairing single-strand breaks?
Primarily, yes. PRP1 is a key player in repairing single-strand breaks and also in base excision repair. Different types of damage than the double-strand breaks we've been focused on.
Okay, so inhibiting PRP messes up single-strand break repair. How does that connect to HRR deficiency being lethal?
Okay, think about it in two scenarios. Scenario 1. A normal cell with fully functional HRR. If you treat it with a PRP inhibitor, PRP gets blocked and single-strand breaks start to accumulate.
That doesn't sound good.
It's not ideal, but it's often not immediately lethal. The real problem comes when the cell tries to replicate its DNA. When the replication machinery encounters an unrepaired single-strand break, the replication fork can collapse, and that single-strand break gets converted into a much more dangerous double-strand break.
So inhibiting single-strand break repair leads to double-strand breaks down the line.
Exactly. But in this normal cell, it still has its fully functional HRR system, so it can use the sister chromatid, which is present during replication, to accurately repair those new DSBs. The cell usually survives. Inhibiting PRP alone isn't typically lethal.
Okay, now scenario 2. A cancer cell with a BRCA mutation, meaning it has deficient HRR.
Right. Now, you treat this cell with a PRP inhibitor. Same thing happens initially. Single-strand breaks accumulate, replication forks collapse, and you generate DSBs. But now, the cell cannot fix these DSBs accurately using HRR because its HRR pathway is broken due to the BRCA mutation. It's lost its high-fidelity repair option.
So the DSBs pile up, or it tries NHEJ.
Both can happen. Either the damage becomes overwhelming and triggers cell death directly, or the cell desperately tries to use NHEJ, which is error-prone and can lead to even more genomic chaos and eventually cell death.
So the combination is lethal. HRR defect plus PRP inhibition kills the cell.
Precisely. That's synthetic lethality. Pathway A, HRR, is defective, but the cell survives. Pathway B, PARP-mediated repair, is inhibited, but that alone isn't lethal. But when you have both Pathway A defective and Pathway B inhibited, the combination is lethal specifically to those cells.
That is just such a powerful concept, and it's why PRP inhibitors have transformed treatment for patients with BRCA-mutated cancers.
Absolutely. It's fundamental molecular biology directly leading to effective targeted therapy.
Yeah.
Understanding this is non-negotiable.
So we have these two pathways, NHEJ and HRR. The cell uses one or the other. How does it actually decide which one to use? Is it just random or first come, first served?
Oh, definitely not random. It's a very tightly regulated decision. The biggest factor, as we discussed, is the cell cycle phase.
Right. HRR needs the sister chromatid, so it's off the table in G1, G0. NHEJ is the only option then.
Mostly, yes. But even in S and G2, when both pathways could theoretically operate, there's active regulation choosing between them.
What controls that choice?
A key determinant seems to be that resection step we talked about in HR, the chewing back of the 5' ends by mRN and CTFP to create those 3' overhangs.
Okay.
If extensive resection happens, it generates the substrate needed for Rad51 loading and essentially commits the break to the HRR pathway. If reception is blocked or prevented, the ends remain more blunt, which favors NHEJ.
So are there proteins that actively block resection to promote NHEJ?
Yes, a very important one is a protein called 53BP1.
53BP1.
53BP1 gets recruited to the sites of double-strand breaks, and one of its key roles is to act as a barrier, limiting or preventing that extensive 5' end resection. By doing that, it effectively promotes NHEJ.
And is there something that counteracts 53BP1 to allow resection and promote HRR?
Yes, this is one of the critical functions of BRCA1.
BRCA1 again.
Exactly. BRCA1 helps to promote resection, partly by antagonizing the inhibitory function of 53BP1. So you have this dynamic balance. In G1, 53BP1 seems to be more dominant, helping to keep resection in check and favoring NHEJ. But as the cell enters S and G2, factors like BRCA1 and CTT become more active, tipping the balance towards resection and enabling HRR.
Wow, so it's like a molecular switch, controlled by these opposing factors, ensuring the cell uses the right repair for the right situation, mostly dictated by the cell cycle.
It's a really elegant regulatory system. The cell cycle state profoundly influences the activity and balance of these pro-NHGJ and pro-HRR factors.
That's fascinating. Okay, let's shift slightly. Thinking about board prep, certain key takeaways always seem to come up. Let's crystallize some high-yield clinical pearls from all this.
Good idea. Pearl number 1. You absolutely have to know the fundamental differences. NHEJ, error-prone, no template, active throughout the cell cycle, but it's the main pathway in G1 and G0.
Got it.
HRR, high-fidelity, error-free, requires the sister chromatid template, so it's mainly active in late S and G2. Get those distinctions straight.
Okay. Pearl number 2.
Know the key protein players for each. For NHEJ, think Q7-ARO80, DNA PKCs, Artemis, and that ligation complex, XRCC4, ligase 4, XLF.
Kids play around loudly.
Exactly. And for HRR, remember mRNA complex, CTIP, those are the receptors. Then RPA coats the strands. Then the big mediators, BRCA1, PALB2, BRCA2, Rad52, helping load the Q recombinase, Rad51.
Right. Memorize those core teams. Now, Pearl number 3.
BRCA1 and BRCA2 are critical for HRR. Mutations equal HRR deficiency. This leads to increased cancer risk, and it creates sensitivity to PRP inhibitors. This connection is tested constantly.
Understood. Pearl 4 must be about PRP inhibitors then.
You bet. The sensitivity of BRCA-mutated cancers to PRP inhibitors is the classic example of synthetic lethality. Understand that concept. Two things that are okay on their own become lethal together. Know it cold.
Okay, and a final pearl.
Pearl 5. Remember that the choice between NHEJ and HRR isn't just about the cell cycle, it's actively regulated. Think about the balance between proteins like 53BP1, which inhibits resection and pushes towards NHEJ, and factors like BRCA1 and CTK, which promote resection and push towards HRR. That regulation adds another layer.
Perfect.
Yeah.
Mastering these 5 pearls really covers the core knowledge here. It's fundamental for RadBio exams and for practice.
Absolutely. And this understanding has immediate clinical relevance. Think about tumor radiosensitization.
How so?
Well, if a tumor cell has a defect in DNA repair, especially in HRR, it's likely going to be more sensitive to radiation. Radiation causes DSBs, right?
Right.
If the tumor can't fix those DSBs effectively, especially using the high fidelity pathway, it's more likely to die from the radiation damage. We actually try to exploit these inherent weaknesses in tumors.
And it applies to chemo too.
Definitely. Response to drugs that cause DNA damage, like platinum agents or topoisomerase inhibitors, is also heavily influenced by the tumor's repair capabilities.
What about the flip side? Normal tissue toxicity.
Hugely important. How well our normal tissues can repair the DSBs caused by radiation is a major factor in determining how much radiation they can tolerate. Differences in repair efficiency between the tumor and the surrounding normal tissues are what give us our therapeutic window.
So understanding repair helps us understand both efficacy and toxicity.
Exactly. And it's moving into the realm of predictive biomarkers now.
Meaning we test tumors for these things.
Increasingly, yes. Looking at the status of genes like BRCA1, BRCA2, maybe ATM, or even components of the NHEJ pathway. This information is starting to guide treatment decisions. It helps select patients who might benefit most from PIRP inhibitors, or perhaps predict sensitivity to radiation or specific chemotherapies.
So personalized medicine based on DNA repair status.
That's the direction things are heading. That synthetic lethality principle with PRP inhibitors is just the beginning.
Right. It's not just a textbook concept. It's impacting patient care right now.
Precisely. And researchers are constantly looking for new synthetic lethal interactions, maybe involving other repair pathways or combining targeted agents with radiation in novel ways to exploit repair defects.
Okay, let's try to apply this. How about a quick mini case? Something you might see. Let's say you have a patient diagnosed with localized pancreatic cancer, and genetic testing reveals they have a germline mutation in BRCA2.
Okay. Germline BRCA2 mutation. First thought. This patient has an underlying inherited deficiency in homologous recombination repair. HRR is compromised in likely all their cells, including the tumor cells.
Right. So how does that knowledge influence how you might think about treating their localized pancreatic cancer?
Well, several things come to mind. First, systemically, even though it's localized now, knowing about the BRCA2 mutation might make you think earlier about therapies effective in HRR-deficient settings if the disease were to spread. PRP inhibitors are approved for metastatic pancreatic cancer with BRCA mutations.
Okay, what about for the localized treatment? Chemo, radiation.
For chemo, pancreatic cancer is often treated with regimens containing platinum agents, like a FOLFIRINOX. Platinum drugs cause DNA cross-links and damage that rely heavily on HRR for repair. So a tumor with deficient HRR might be more sensitive to platinum-based chemotherapy. That's a key consideration.
Interesting. And for radiation?
For radiation, it's a bit more complex. On one hand, HRR deficiency should make the tumor cells more sensitive to radiation-induced DSBs. That's the theory. But you also have to consider that the patient's normal tissues also carry that BRCA2 mutation and are HRR deficient. While normal tissue response is complicated, there's a potential concern for increased normal tissue toxicity. So it might make the tumor more killable, but you need to be very careful about the dose and delivery to spare surrounding organs. It adds a layer to treatment planning and toxicity management.
So knowing the BRCA2 status doesn't give a simple yes or no answer. but it provides critical biological context for weighing the risks and benefits of different options, especially platinum chemo and potentially radiation sensitivity versus toxicity.
Exactly. It's a perfect example of how understanding these fundamental pathways translates directly into nuanced clinical decision-making.
Great example. Okay, let's switch gears again and lock this down with a quick board blitz.
Yeah.
4 questions, multiple choice format. Ready?
Let's do it.
Question 1. Which of the following is a characteristic of non-homologous end joining, NHEJ, that distinguishes it from homologous recombination repair, HRR? A, it's an error-free repair mechanism. B, it requires a sister chromatid as a template. C, it is primarily active only in the S and G2 phases of the cell cycle. D, it can lead to small insertions or deletions at the repair site.
Okay, distinguishing NHEJ from HRR, NHEJ is the error-prone one. So D, it can lead to small insertions or deletions. A, B, and C are all characteristics of HRR.
Correct. D is the answer. NHEJ's lack of a template leads to its potential for errors.
Makes sense.
Question 2. Which protein complex is crucial for recognizing broken DNA ends and initiating the NHEJ pathway by recruiting DNA PKCs? A, Rad51, B, RPA, C, Q708 Eddy heterodimer, D, CTIP.
Initiating NHEJ, the first responders.
Right.
That's Q, the Q7080 heterodimer. So C.
Correct again. C, Q7080 binds the ends and brings in DNA PKCs. Rad51, RPA, and CTIP are all HRR players.
Got it.
Question 3. A cancer patient with a germline mutation in BRCA1 is likely to have impaired function in which DNA repair pathway, making their tumor potentially sensitive to PRP inhibitors.
Right.
A, base excision repair. B, nucleotide excision repair. C, non-homologous end joining. D, homologous recombination repair.
Okay, BRCA1, that's essential for HRR. Impaired HRR leads to PRP inhibitor sensitivity. So D, homologous recombination repair.
Yeah, so D is the answer. BRCA1 is key for HRR, and its loss drives the synthetic lethality with PRP inhibitors.
Right, classic link.
Final question number 4. The selection between NHEJ and HRR for repairing a DNA double-strand break is significantly influenced by the cell cycle phase. HRR is predominantly active in A, G0 phase, B, G1 phase, C, M phase, D, late S and G2 phases.
HRR needs the sister chromatid template that's only available after replication, so late S and G2 phases. Answer is D.
Perfect. D is correct. HRR activity is tightly linked to the presence of the sister chromatid in late S and G2. NHEJ handles things in G0 and G1.
Good review.
Excellent. See how focusing on those key distinctions, fidelity, template, cell cycle, key proteins, helps you navigate these questions? Understanding the why is key.
Definitely. And thinking about that regulation, that balance between 53BP1 promoting NHEJ and BRC1 promoting HRR, it really sparks ideas about where the field might go next.
Yeah. What kind of questions does it raise for you thinking about future therapies?
Well, considering how delicately that pathway choice is balanced, especially around that resection step, what if we could develop drugs that specifically target those regulatory proteins? Like, could we force tumor cells, maybe ones that aren't fully HRR deficient but rely on it somewhat, to only use NHEJ?
Deliberately push them towards the error-prone pathway.
Yeah. Could targeting something like CTIP to block resection or somehow boosting 53BP1 activity make those cells more susceptible to damage? Could we essentially create an NHEJ-dependent state that's more vulnerable to radiation or certain chemos?
That's a really interesting thought. Manipulating the pathway choice itself as a therapeutic strategy, rather than just exploiting existing defects.
Right.
It definitely speaks to the cutting edge of DNA damage response research.
Absolutely. Finding ways to tip that repair balance therapeutically. There's still a lot of potential there.
For sure. Okay, let's wrap up this session. We've covered a lot of ground on the cell's 2 main ways of fixing double-strand breaks.
We hit NHEJ, the faster, error-prone pathway. Works all the time. Uses Ku, DNA PKCs, Artemis, Legates 4 complex.
Kids play around loudly. And we covered HRR, the slower, high-fidelity pathway, needs that sister chromatid template in late SG2, relies on MRN, CTP, RPA, BRCA1, PALB2, BRCA2, and Rad51.
We spent time on the critical importance of BRCA1 and BRCA2 for HRR, how mutations cause HRR deficiency, increase cancer risk, and create that synthetic lethal vulnerability targeted by PRP inhibitors. Huge clinical relevance there.
And we touched on how the choice between these pathways is actively regulated, influenced by the cell cycle, and that key balance between proteins controlling DNA and resection, like 53BP1 and BRCA1.
It's a lot, but mastering these pathways really is foundational for radiation oncology.
Absolutely. Keep reviewing these concepts, keep building that knowledge, you're putting in the work, and you're definitely on the right track to excel in your exams.
And next time, we'll keep building on this foundation. We'll explore another crucial area in RadBio, looking at the cell cycle gatekeepers, P53, P21, and the RBE2F pathway.
Sounds great. Remember, you can find more resources and complete practice oral boards over at radonsmartlearn.com.
That's radoncSmartLearn.com. And make sure you subscribe to Rad Onc Smart Review so you don't miss our next session.
Thanks so much for tuning in.